Our next speaker, Dr. John Yarbrough, is a physics researcher at the National Renewable Energy Laboratory. Dr. Yarbrough's research interest lies in the development and implementation of novel characterization techniques for imaging and spectroscopy. His talk this morning will be on the use of super high optical resolution microscopy to probe the interaction zone between Clostritium, thermocellum, and biomass. It looks like we can see your screen. Can we hear you?
Yes, I hope you can hear me.
Yes, we can hear you. Go ahead.
Cool. Yeah, thank you for that introduction, and thank you for the EMSL Integration 2020 Committee inviting me to give this talk on leveraging super high resolution optical microscopy. We work in the field of bioenergy, moving away from other viruses and bacteria that we've seen previously. And what our goal is within the CBI organization funded through DOE, is we want to take the plant side, do a lot of characterization, genetic mutation to improve the plant's ability to be degraded down to biomass, to sugars, as well as we want to look at the microbes, and in this case, the anaerobic thermophilic microbes that actually break down that plant material, come together and do a consolidated bioprocessing and co-treatment to get all the secondary products that we want to generate fuels. And what we're interested in my area that we're working on is the Clostridium thermocellum bacteria cell. And the reason why this is important is that C-therm is so far the most efficient cellulitic bacterium that has been isolated. And C-therm can solubilize more than 60% of the C5, C6 sugars within a 4 to 5 day period from non-pretreated biomass. Normally we use pretreated biomass using chemical pretreatments to make it easier to digest. C-therm does not need that. And what we can see here is that with the fully intact microbe, we get a significantly larger glucan conversion as compared to free fungal enzyme systems. And we associate this with the cellulosome from the C-therm. And this is a rather complex tethered enzyme that appears to be in the presence of the microbe on the surface of the microbe. And yet, when it's by itself, if you just separate the cellulosome from the bacterial cell wall, you do not see the same conversion. So there's something going on that you need both the cellulosome and the bacteria working together in order to achieve these high conversion rates. The question is why? And if you look here we actually have transmission electron microscope image. Here we have the bacteria. Here we have the plant material, the plant cell wall. And then in between the bacteria and the plant cell wall is that we've got this fuzzy looking material, which we are going to define as interaction zone. And the question becomes is this just simply peeled lamella from the plant cell wall, or is it actually cellulosomes or a combination of both? And this is the area in which we truly believe is the reason why we see such high efficiency and conversion of glucan with the microbe. And if you look at the 3D representation, this is the area in which we are defining as the enzyme microbe substrate interface. We want to get a better image and a better idea on what's going on there. And to do that, we have several optimal techniques that can be used to study this microbial surface interactions. One of them is optical imaging. The beauty of optical imaging is that you can get large areas of field of view, numerous number of bacterial cells, but the resolution, you're stuck to the diffraction limit of light, which is typically above 250 nanometers. You can then move into scanning electron microscopy. It gives you far higher resolution, up to about 10 nanometers. You have the ability to actually look at the more ultrastructure of the bacteria. Here you can actually see that the distal ends of this Clostridium thermocellum are rather textured, and the middle is rather smooth. You can also move into TEM, which gives you far more superior resolution, finer detail, and can get us down to resolutions less, about 2.2 nanometers. The issue we have is that with these electron microscopy techniques, there is a significant amount of sample prep that is involved. You take the bacterial cells out of their natural environment, you subject them to sample prep, dehydration, and bedding just to be able to do that. So now you've gone from a native environment down to very sophisticated, complex sample prep that puts them then into a vacuum atmosphere to try to do the imaging. So the question is, how can we get past without using electron microscopy? Can we use optical techniques to push us beyond the diffraction barrier of light and closer to the actual resolution that we see with electron microscopy? And we can. And that's through the techniques of the past 10 years of super high resolution microscopy, which I'm not going to go into how it works, but how we utilize the super high resolution is that we actually have the ability to take the wild type and generate different mutants. And with that, we have two probes. One is a photoactivatable GFP with a type 1 dockerin, which has the ability to adhere to an empty slot on the SIP-A scaffold or the cellulosome, as well as an open slot on the OLPC site of the bacterial cell wall. And we also use an Elexafluor 647 that is specific to the CBM3 binding module, or the CBM3 that is located on the scaffold of the SIP-A. And each scaffolding, each cellulosome only has one CBM3. Therefore, any fluorescence we get, we know is coming from a single cellulosome scaffolding. We have the ability to do genetic modification, in which we can actually remove the primary scaffolding and allow free cellulosome systems to mimic that of what we see with just pure cellulosomes. And both of these systems allow us to do the blinking, allows us to get that super high resolution microscopy techniques accomplished. And what do we see with super high resolution imaging? Well, back in 1987, Ed Baer looked at Clostridium thermocellum, and he's noticed what he defined as polycellosome protuberance, identified back in 1986. And this is what we see with the yellow circles, is that these round oval type shapes on the surface of the bacterial cell wall, he associated it with these cellulosome clusters through secondary antibody labeling. And as he presented this work, one of the questions came are those real? Is that an artifact due to the staining procedure? It was still questioned what those were. People were somewhat skeptical if they were polysomal protuberance. Well, 30 years later, we take the same bacteria, run it through super high resolution, and what we notice is when you tag the cellulosomes with the CBM3 and the Elexafluor 647, we see very similar structure on the outside of the bacterial cell wall, signifying that yes, what he was seeing back in 1987 is in fact these polysomal protuberance, which is a high concentration of cellulosomes decorating the exterior of the bacterial cell wall. And we can see in the optical image, it actually decorates, twists around the bacterial cell, and that bacteria is heavily decorated with these polycellosome protuberance. And they're very similar in the structure of Ed Baer. Now we have the ability to actually image both single cells and the bacteria on actual biomass. In this case, we're looking at AVICEL. And here what we're looking at is here you have some individual super high resolution storm imaging of the bacteria and an image of the bacteria actually interacting with the AVICEL. Here we look at the zoom up close up image, and we can actually see the outline of the bacterial cells, and we actually see an increase in fluorescence at the distal ends of the bacteria where they're attaching to the AVICEL particle. As we zoom in, we can start seeing the power of this optical resolution. We are achieving going beyond the diffraction barrier of light. And if we look closely here, you have an actual bacteria cell wall, and you can start seeing these small circular spheres. Those are actual fluorescent events that we are associating with the CBM3 and the Elexafluor tagged cellulosome. And what we see is that here we have a string of 4 spheres, which are separated a total of 130 nanometers. And we have the ability to actually visually separate these fluorescent events. We come over here, we see they're a little further away, 230 nanometers, and we're seeing easily that we're getting 6 nanometer separation beyond the diffraction barrier of light. So this has proved to be a very valuable tool and be able to characterize the distribution of these cellulosomes on the bacterial cell wall. The issue is with all optical microscopy is that we really need to be able to quantify this information. And the reason why is here we see cellulosome grown on cellobios, and we capture it in the log phase. And as we can see, the bacterial cell wall is heavily decorated with these cellulosomes. Yet, as the bacteria propagate and transition from log phase to stationary phase, visually we see a decrease in the overall fluorescence of the bacterial cell wall. And the question is why? Is this a real effect, or is this an artifact due to the image processing, the way that we're running it through ImageJ? Because you know, whenever you're in an optical imaging software package, you can actually adjust the backfill, the brightness, the contrast. And so is this just a simple collection? You didn't have the right PMT setting, and that's the reason why they're different, or is this an actual real effect? We see the same issue when you actually looked at avocell and the bacterial cell wall, and the bacteria on the actual avocell. And what we have here is, here we actually have highlighted in the red boxes is avocell particles. These bacterial cells have reached stationary phase. And as we zoom into this particular avocell, we can clearly see the outline in the white light image of the bacteria, and we can see the fluorescent patterns. And as we zoom in and only look at just the fluorescent patterns, we do see an increase in fluorescence at the distal lens where the bacteria is attached to the avocell. And we do see fluorescence running all along this avocell particle. But is that really an increase in the number of cellulosomes, or an issue with the brightness and contrast? And as we zoom into this particular bacterial cell, you can see clearly at that distal end where the bacterial cell wall is connecting, adhering to the avocell particle, we do see an increase in fluorescence. We want to be able to characterize that. And over here, what we have is that basically reminds you where the green fluorescence is coming from, as well as the red. Red is coming from the specific SIP-based scaffold. Green is either, in this case, is coming from an empty slot on this SIP-based scaffold. And all of these smaller GFP fluorescent green are open OLPC sites on the bacterial cell wall. And so with this technique, we actually have the ability to characterize that, and we'd be able to see this actual overall increase in fluorescence. But is that real, or just an artifact of the way we pick the images? That's why we moved from simple just image analysis, doing line cuts, looking at the overall intensity, comparing that, to actually using machine learning algorithms, in which in this case we're using a density-based spatial clustering application with noise, or dbScan. And what dbScan is an unsupervised machine learning referred to as a cluster analysis. It propagates cluster detection method, linking points that are closely packed together. This removes any bias from the user, and we allow the machine to do unsupervised and find its own clustering. And so take, for instance, here we have some fluorescent events that we're going to find as a CBM3 molecule. And what we use the dbScan, and it can detect outliers using defined epsilon radius and a minimal number of points. And as the algorithm goes, based off of the criteria we give it, it can detect arbitrary shaped clusters and is quick and robust to outliers. And so what you see here is that it's identified all of these as part of the cluster. It's able to identify the edge of the cluster, which is shown in yellow, and can actually identify what we assume is noise and ignores that. So it's able to actually define arbitrary shaped clusters based off of a user-defined epsilon radius and a minimum number of points or fluorescent events, fluorescent molecules. And what we chose to do was to set the epsilon to 75 nanometers, which gives a diameter of 150 nanometers, which is typically the full length of an extended cellulosome. And we ranged the number of points from 10 molecules, 10 cellulosomes within this epsilon to 1,200, sometimes even larger. That way we can now start seeing clustering, higher density of packing of cellulosomes, which in theory would give us a higher increase in fluorescence. So when we go back to that image I just showed of the avocell particle, the beauty of the super high resolution technique is that it actually spits out a raw data file, which gives you the XY coordinates of each fluorescent event. That allows us to be able to separate the CBM3 channel, to be able to only look at only the fluorescent events associated and their location associated with the CBM3. And what we see here in this image is that, yes, we see more number of cellulosomes in the area next to the bacterial cell wall as compared to other parts of the avocell. We can look at the GFP labeling, so now we can see where that photoactivated GFP is located on the bacteria, on the avocell and bacteria. and we know the precise location when we combine it. So now we're no longer looking at being able to adjust the intensity or the brightness. We're looking at actual physical location of these molecules. We then flow it into the dB scan analysis. And what this starts showing is, here I show the cluster analysis generated by this algorithm. And we'd identified 49 clusters with having at least 500 molecules per epsilon, saying that within that defined radius, at least 500 molecules are within that area. And what we see here with the CBM3 is that, yes, at the point where that bacteria is adhering to that cell wall, we have a significantly increased clustering of the CBM3, which says that that is actually help tethering that microbe to that bacterial cell wall. We also see rather large areas that extend further away from the bacteria. So here you have the bacterial cell wall, it's adhering to the avicell particle, but you see this large cluster extending farther away from the actual bacteria, signifying that as that bacteria attaches to the avicell, it's actually shuttling cellulosomes from the bacterial cell wall onto the avicell particle. Here we're actually looking at the, so here we identified 27 clusters on the actual bacterial cell wall. We also identified 22 clusters located on the avicell particle. And this is the power and the beauty of this technique, is that now we can start quantifying this information. And what we see is the observations is, if we look at the photoactivated GFP type 1 doctrine, we just simply see an exponential distribution. It starts high with very small clusters, with a cluster with a very small amount of molecules, and that number of clusters decreases as you increase the minimum number of particles. But what you do see is a different distribution pattern when you look at the cellulosome distribution. What you see here is that when the bacteria is interacting with avicell, you actually see higher number of clusters with higher number of molecules per epsilon, signifying that when they're attached, they're generating a ton of cellulosomes that are binding to the surface and helping degrade that material. And so this is what gives us the power. So here what you can see is that even at higher clustering, around 1,500 molecules per epsilon, we are still seeing about 10 clusters that we were able to identify, showing that these cellulosomes can be highly packed within the surface of the avicell as well as the bacterial cell wall. And so, like we said, we see an increased number in larger cluster size, interaction zones, and large clusters. And here what we see is, when we look at just the cellulosome in log phase and stationary phase, do we see a difference? And yes, what we actually see is, as we increase the number of molecules or the clustering algorithm, we're seeing that, yes, during and stationary and log phase, we are clearly seeing higher number of clustering of these cellulosomes on the bacterial cell wall, showing that as they enter stationary phase, they are shedding off those cellulosomes. So if you look at the initial scans in individual cell BIOS, we looked at 65 individual bacterial cell analyzed in log phase that were grown on cell BIOS. We looked at 70 individual bacterials on stationary phase. And what we saw here, clearly we see a difference in the distribution pattern of the cellulosomes on stationary versus log phase, because we're able to now look at multiple bacteria and look at the overall average. And what this shows is that you have an exponential distribution versus this rather distribution. It shows that, yes, during stationary phase, there is more cellulosomes on that surface. And if you look at avicell, at the single bacterial cells, here we looked at 73 individuals in log phase, 62. And we clearly see two distinct different distributions, that as you enter log phase or stationary phase, you see that shift from a nice distribution pattern here to an exponential distribution pattern. Same with avicell, you see the reverse. With avicell particles, you see a log phase, you see a rather exponential distribution. As it goes to stationary phases, that bacteria is shuttling the cellulosomes onto the surface of the avicell. And now you see a difference in the distribution pattern of the clustering. So you're seeing higher number of molecules per cluster on the avicell particle. And here we're able to look at 6 individual particles, both in stationary and 8 in stationary phase. And so this gives us the ability to quantify and truly start understanding the distribution pattern of these cellulosomes and why we're seeing an increase in the synergistic effect of the bacteria. And so what this study is starting to show is that while we look at the polysomal protuberance of the CBM3, while in log phase, we see larger number of clusters with molecules per cluster. We see in stationary phase, they're losing those cellulosomes. There's a clear difference in how the bacteria looks between log and growth phase. And we're seeing shuttling onto the avicell particles, signifying that as they're attached, they're dumping a ton of cellulosomes onto that surface, which helps degrade that material. And with that, I would like to thank our main funding source, the U.S. Department of Energy CBI program, the University of Colorado School of Anschutz. That's the facility in which we do all this microscopy imaging, and our task leader, Yannick Baumel, and all the co-workers that helped us on this project, including Dartmouth and Ed Bear.